Good morning. Okay, so today we are continuing with the genetics packet. I labeled the bottom of my pages, so we're looking at the bottom of page 5 right now, and we're going to be doing some problems which are called multiple alleles that you guys may know more as blood types. So you should have a page within your packet. It actually says multiple alleles on top, but then it starts out with some blood type problems. On the back of that, I've written a few notes that you guys can go through and look at as well and probably copy through. They'll be on Google Classroom if you haven't gotten there. What you guys should remember about your blood types is that there are 4 different types of blood, and your blood types that you see here are actually going to be your phenotypes. So you have some people who are blood type A, some who have blood type B, some have blood type AB. So if we look at just that right away, blood type A is dominant, blood type B is dominant, but together they are co-dominant, meaning that A is not dominant over B is not dominant over A, and we kind of get them shown together at the same time. Your 4th type is blood type O. And blood type O is our recessive type. So we could say type A is dominant over type O. Type B is dominant over type O, making type O recessive. So we kind of have this co-dominant problem as well as a monohybrid cross problems almost put together within our keys here that you guys will see. So the difference between these particular blood types is the protein that's located on the outside of your blood cells. So we're just using this little diagram as a representation for you guys. So let's say this particular arrow is the protein that we find on the outside of blood type A and the people who have that type of blood. If we're looking at people who have type B blood, they have a completely different protein. Meaning, if you are a type B person and you get a blood transfusion from a type A person, this protein gets put into your body's going to try to fight it off right away, and that actually will end up killing you by the time it makes its way through your system and your organs and everything begins to shut down because your body's not recognizing this particular protein. Same thing if you put B into a type A person. When we look at type AB, however, they have both the A protein and the B protein. So because of that, they can kind of receive type A blood, they can receive type B blood, they can receive blood from a type AB person. They can also receive from a type O person, because when we get down to type O does not have any proteins that are recognized on the outside of that red blood cell. So for that reason, we can give type O blood to anybody. If you're type O, you can get it. If you're type A, I have no proteins that it recognizes. Same with B, same with AB. For that reason, type O person is said to be a universal donor, which means that they can give their blood to anybody. About 85% of the population does have type O blood. If we go back to a type AB person, about 1% of the population have it, but they are the universal recipient, which means that they can get from any blood type. It doesn't matter what blood they get put into them. Their body recognizes the A protein, the B protein, the ABs together, as well as the no protein that's on type O. Type B is about 4% and type A is about 10% of the population. So we're looking at these particular phenotypes on the front side of this particular page when we look at the problems that you guys will be doing. And the thing about it is that you guys are still going to be doing all of the same steps. So from first year bio, you guys might remember 2 different methods in order to do your particular key. And the good news about this, your key will always be identical. You're always going to have 4 parts to the key. So type A, type B, type AB, and type O. The more scientific way to do it is using your big I's and your little i's. And we usually use the letters, the A's and the B's, as your sort of superscripts here. So if I said, okay, well, type A blood is dominant over type O, we can say type A blood is big I A. That's my homozygous dominant type A. Well, since it's dominant over O, I can also be heterozygous. Big IA, little I. And that little I represents the recessive choice. So if we look at blood type O, it is little I, little I. It's recessive, so it gets the little or lowercase i's there. Type B is dominant over type O. Big IB, or big IB, little I. Type AB, it's a mixture of the 2. We see both at the same time. So we take one from big IA and one from big IB. So only one choice here. You can't have homozygous dominant. You can't say I have heterozygous. I only have one choice here. It's co-dominant. So that with the I's is your more scientific method way, or some more scientific way of doing it. First year bio, and we're fine if you guys want to write them out this way, is using A's and O's. So we say that big A is my dominant, big B is dominant, little o is recessive. So type A could be big A, or big A, O. So there's my homozygous choice, my heterozygous choice. Type B can be big B, or big BO, same thing. Homozygous dominant, heterozygous. AB is only one choice. I'm taking one from here, mixing them together. I have AB. Type O is little o, and that's still my homozygous recessive. That is your key. I don't care if you're doing it with the I's or you're doing it with these, but I need to see one of those two with my 4 choices that you guys see there. From there, we want to write out my parents. So my first cross, so step 1, here's step 2. My parents are type AB cross with a heterozygous type A parent. So depending on which method you do it, here's my type AB, big IA, big IB. Here's my heterozygous type A, big IA. There's that little I there. Same thing here, big A, big B, cross with AO. They mean the same thing, depending upon which key. I don't need to see both versions there, though. Then you guys go through and do your Punnett square. One parent across the top, one parent across the side. You guys know how to do this. You guys know how to drop them in and fill them in. Keep your A's before the B's. Keep your capital letters before the lowercase letters. Same as what you've always done, whether you're doing the I's or the AO's. Once you guys get the genotypes and phenotypes, they're going to start to get a little bit longer. So my genotypes, I have a lot of choices here. So whether you're using the I's or the A's and O's, fill them all out. So I big A, or I have I big A little I. Those are my 2 type A's, but you have to go through and fill them in. So when you look back, I've got one choice here. I've got one that's I big A little I. You do the same thing with the B's. Write out both choices. Homozygous dominant, heterozygous. I have one choice for AB. I have one choice for O. So write them out genetically. What do they look like? I should have 4. So I have a 1 to 1 to 0 to 1 to 1 to 0 ratio there. You guys want to do the same thing with the phenotypes. So physically, what type of blood do they have? I know my first 2 here are blood type A. My second 2, big IB, big IB, big IB little I is type B. AB is this one, and then I have my O. Write out my blood types. What do I have? It should equal 4 there. So if I cross those 2 parents, my possible children could be, I have 2 that could be type A. I have 1 that's type B. I have 1 that's type AB. It doesn't mean that these parents are going to have exactly 4 kids and this is what they're going to look like. It means I have a 50% chance of having a baby that has blood type A. I have absolutely no shot in the world of having a baby of blood type O. Basically what you guys are looking at there. So you guys should now be going through, trying those little problems there. Once again, depending upon which key you guys want to use, but go ahead and try those particular problems. See what you guys can do with them. There's a little practice problem at the bottom there. We'll make sure that if you have any questions, you guys can check with us on those. Continuing in your packet, you guys are now going to continue the next couple pages. So like I said, I've numbered my pages. Now that you've finished your incomplete dominance as well as your codominance, we're now on part 5, your multiple alleles. I ask you some questions and then search the problem. So you guys are finishing page 5. You guys can do all of page 6. It asks you about showing all 5 steps for these and determining, kind of answering a little question here as well as down here based on the information that you get from completing those Punnett squares. You guys are going to continue on to page 7. Page 7 is going to show you guys an RH factor. So I want to go back and kind of review that. RH factor is another protein that you would find on your blood. So besides being blood type, you also have an RH factor on that blood. An RH factor is just like a regular monohybrid cross. We can say that if you are RH positive, or you could be RH negative. RH positive blood is dominant over someone who is RH negative. It means that you have no additional proteins there. So if I'm RH positive, I have a different protein, and here I would have no proteins. This is different than our blood types. So for instance, I could say I am type AB positive in terms of my blood type. So blood type and then the extra protein, which is that positive factor. Regular key, I can say if I'm homozygous dominant, plus plus, or I can be heterozygous, plus minus, or plus negative there. RH negative is negative negative. So here is my dominant choices, here is my recessive choice. It is like a regular monohybrid cross with that. Hope that helps. Any questions, please let us know. Continue working your packet. Don't slack off on this stuff. Due dates will be posted later in the week. Thanks.